Alrighty. Good morning. Good morning, good morning, good morning. It's Wednesday. It's sunny. It's another sunny day. And we're going to talk about things more excretion. Thank you for those of you. There's the Freudian right there at the top. And let me clear this. Your wonderful words. And let's consider where we're going by considering where we've been. We spent the our last time together talking about circulation. We're expanding. It was like part 2 of our respiration time together. So we talked about circulatory systems. We moved away from thinking about fusion and Fick's law a week ago today, and then we talked about circulatory systems open and closed. We talked about different respiratory pigments, and then we considered one of the most reduced and confusing and taxonomically tortured of the metazoans, the mixozoa, which has lost their mitochondria entirely and really does not seem to have a need for oxygen, which I do, and most of the metazoans that I know, we all do. But if the mixozoan don't care. What are we going to talk about today? We're going to move along a line of, I suppose, consequence. We're going to talk about what are the consequences, how do organisms, does the vast diversity of invertebrate organisms, how do they deal with consequences of respiration and metabolism. And so some of these, specific to metabolism, we're going to talk about nitrogenous wastes and about how organisms have adaptations to deal with the excretion, with voiding themselves of nitrogen. We'll talk about 4 major categories of them. We'll talk about some classic and maybe some new, but some classic excreting organs. And then we'll talk about them as we see them in the landscape. And as that landscape time through evolution, so the ecology and the evolution, we'll talk about strategies of conformation or regulation and what that means for organisms, particularly in freshwater or a marine environment or the intermediate like brackish water. In talking about this lecture in the past, I had talked about this with my 2 sons, and they figured that instead of calling this lecture 9 excretion, last year they thought it should be called Everything Everywhere is Peeing, and sometimes it matters, even to my stinky kids. So everything everywhere all at once is peeing. Sometimes it matters. My hopes for you today is that you'll be able to distinguish between different forms of nitrogenous waste, different forms and organs of excreting that, and then be able to go out onto the intellectual and knowledge-based environment to appraise, synthesize, and interpret how those relate to invertebrates in the world about questions of diversity and ecology. So, when we're talking about excretion, we are talking about the removal of, in this case, different amino acids and different byproducts of protein. So removing waste from the body, and it's critical towards maintaining a saltwater balance, and it's critical towards filtering out toxins from the body, because when we consume proteins, the byproduct of that is that those proteins become deaminated, and we've got amino acids floating around. And one leftover part of that process is ammonia. And ammonia is very, as Brittany would say, toxic. There's your earworm for the day. Now, it's not the only kind of byproduct that's produced by metabolisms of proteins, and it's not the only, the toxicity is not the only problem. So ammonia is not the only one. The toxicity is not the only problem. There's also associated problems, osmolarity problems, so losing water, and this is particularly important if you're in an environment where water is in short form, like if you're living terrestrially. So when we're thinking about nitrogenous waste, we can think about, they're all toxic, but ammonia, urea, uric acid, and guanine are 4 of main principal categories of these nitrogenous wastes. So ammonotelic organisms are ones that excrete ammonia. And that ammonia comes from the catabolism of proteins and amino acids, and it's really toxic. And that's principally because ammonium ions have a potassium ion-like behavior, and so it can really affect the membrane potential in nervous systems across cell walls. Now, luckily, I suppose, it's really soluble in water, and so it's an ideal waste product for aquatic animals. So that high toxicity is really, it's diluted in aquatic habitats where there's plenty of water to dissolve it and to disperse it away from the body. So there's kind of an amuse-bouche of, we see crustacean arthropods, platyhelminth, flatworms, cnidarians, porphyrians, echinoderms. So this is not like a monophyletic group of monotelic organisms, but largely described by where they live in an aquatic. So on land, and like, again, with our biases showing, this is one that we're pretty familiar with. So we're a ureotilic organism. We convert ammonia afterwards into urea, which isn't as toxic, and it doesn't require as much water to excrete it. And that's generally why we, as terrestrial organisms, tend to use it. Now, there's another form that's used by you might have come across because of your familiarity with birds or reptiles, and that's a uricotelic organism, which excretes excess nitrogen as uric acid. That's more energetically intensive than converting ammonia to urea, but it changes it to something that has it's low toxic, and it's not as soluble in water. And it tends to be used, again, in terrestrial environments where water loss is a concern. So we see it in terrestrial snails and slugs. So within the phylum of the mollusks, we will see some of them being monotelic and some of them being uricotelic, but the uricotelic tend to be the terrestrial snails and slugs. And the insects. So, of course, the vast diversity, species diversity of invertebrates living on land. And then closely related to uricotelic ones, we see some that convert uric acid to guanine, which is low toxicity, low water solubility, tends to be excreted in a solid form, and phylogenetically, we tend to see it within, most prominently within the arachnids, so within the arthropods, the chelicerates, and the arachnids. And I'm rearranging my windows here below to see who's in the waiting room and to see if there's any questions right off the bat, but it doesn't look like there is. So in considering these four major kind of principal players in the play of excretion, it seems you are familiar with those. So hooray. A quick review. Now, when we broaden out to think about strategies and organs, we're going to talk about nephridia, metanephridia. We'll talk about renal sacs. We'll talk about coxal glands. We'll talk about Malpighian tubules, and we'll talk about green glands in a whole bunch of different organisms. But the first thing to consider, so the nephridial organs up here, we'll think about nematodes, but like the flatworm is kind of a classic example that I'll show you. The antennal gland, or this green gland down here, that's our crustacean example. We have renal glands, and this one is a mollusk, but a cephalopod mollusk. And then for some chelicerates, we have a coxal gland, and the Malpighian tubule is an insect example. An insect example that we'll dissect, like we'll talk about, but of course you might remember from 2700 that we've seen Malpighian tubule-like organs that have independently evolved in different arthropod lineages that have made the successful move to the terrestrial environment. Now that first category, though, sponges, cnidarian, ctenophores, echinoderms, no specialized organ. So what's happening there? So with the ctenophores on the left or the cnidarians on the right, they're excreting ammonia through diffusion largely because their body fluid is so similar in composition to seawater, and so it's kind of going away. That byproduct is diffusing away. Now, if you think about ctenophores and cnidarians from a done-it-all perspective, we're talking about some of the earliest branching animal lineages, where we don't tend to see. We've talked about them when we've codified that part of the phylogeny. We've talked about it as organisms lacking symmetry, and then we talk about moving into the parts of the tree that are bilaterally symmetrical, but also we've called it the nephrozoa. Now, the nephrozoa as a word, as a systematic word to incorporate all of the organisms with an excretory capacity. That's me tying the phylogenetic into the functional that we're talking here. So the nephrozoa here, we see, so the nephrozoa, this would include, as the diagram might make you think, it includes all of the spiralians, all of the deuterostomes, all of the ecdysozoans. So us and all the worms and all the insects and all of the other paraphernalia of those regions. And the outgroup to the nephrozoa that was resolved with the resolution of the mystery of the purple sock was the xenocoelomorph. Remember, the kind of like the purple socks that littered the deep ocean off Monterey Bay, California. So we just finished talking about cnidarians or ctenophores and about how there's really just diffusion that's taking care of these byproducts. And so once we get into the nephrozoa, that's when we start to see specialized excretory organs, specialized excretory tissues and cells that are actively transporting and might also be using ultrafiltration. We'll talk about that with the renal sac and in the crustaceans as we go along here. Sorry, with the mollusks as we go along here. But it seems a couple of years ago that we might actually have to add, here comes a mute, I'm going to clear my throat. I hope that didn't make it through. But when we're talking about the nephrozoa as being the excretory organs and active transport, we might have to add xenocoelomorphs as the sister taxa to the nephrozoa, not having specialized organs for excretion, but using active transport, because we've learned, as that second bullet says recently, that xenocoelomorphs appear to use active transport and diffusion to concentrate excretory products. And that most of the genes that we see that are involved in excretion in the nephrozoa, in the vast morass of this diversity, are already present in the xenocoelomorph, and some of them are present in non-bilaterian animals. So using the phylogeny, using our phylogenetic perspective as a way to calibrate, it's our GPS to calibrate our movement through the function of all of the properties that we're moving through in this course. Now, the nephridia we talked about in our shared time together in 2700, and we called them, remember the common name for them would have been a flame cell. And if you don't remember why we called them the flame cell, I'm going to show you a video in a second, but it's essentially what we've got here. So left to right in this diagram, we've got the flatworm. So unbisected, it hasn't run into the Nobel laureate with the sharp scalpel that liked to cut them all into tiny, tiny pieces. But we see this protonephridial network lining the outside margins of our flatworm here. And if we blow that up, when we take a look at these little tip ends, those are the nephritophores. And then the nephritophore, if we look at it in cross-section, so we are going to use our scalpel, I suppose, but only in a diagrammatic way. If we take the long conducting tubule all the way to the bottom, we see this flame cell. And if we blow the flame cell up, that's what we have here. And this flame cell has two flagella that sit in this nuclear cap cell. And they are doing what do flagella do? They flagellate, right? So this end, the nephritophore, opens into the body cavity and opens at the other end to the exterior of the body. And the main excretory product there for these is ammonia, and there's a classic animation that I like that was simplified a few years ago by some instructors at the University of Alberta here. So you can see the flame sitting inside the body, and the beating of that flagella in this case just one shown in the diagram, that is drawing towards it all sorts of fluid that contain amino acids and sugars and ions and wastes and hormones and water. And so as that pressure change is created by the beating of the flagella, they then have, so the water flows in and then along the long nephridial or protonephridial tubule. And the cells that line this, their responsibility is essentially to reclaim amino acids, to reclaim sugars and ions and waters that might otherwise be excreted, and to make sure that the excretion is really, what do we have here? Red is our excretory project, is that it moves out. And why is it called a flame cell? And because when we looked at it under a dissecting microscope, this is what it looks like. And if you spent, for those of you who spent time with flames staring at them, that's what this reminded early microscope using invertebrate zoologists of. So with the flatworms, with the nematodes, the metanephridia, and the protonephridia are some of the specialized excretory organs. So again, there, sometimes in the past we've talked about, with respiration, we talked about the appearance of respiration being contingent or coincident with size. Now here with excretion, with the toxicity associated with excretion, we see our organ can be much smaller. So this nephridia and protonephridia, these are not big things, right? Like this is we need to look through a microscope to see it. And so the organisms that possess it are slightly bigger. We wouldn't see a circulatory system in a nematode, so we don't see a specialized organ associated with that in the nematode. But in the nematode, we do see specialized organs associated with excretion. Now, let's switch. We'll change the channel. This is the Gen X person. I'm changing the channel so you can use your remote. I'm going to use this, and we'll adjust the UHF and the VHF, and we will take a look at the molluscan channel, and we'll specialize to cephalopods. And you've seen this before when we talked about cephalopods in 2700, but this is a reminder that in cephalopods, where they're excreting, it's principally via the gills here, right? These large gills. And remember, with the closed system, circulatory system of the cephalopods associated with that active, fast molluscan lifestyle. But we're going to back the story up slightly within the cephalopods, and I'm going to call your attention to the red, which is the flow of blood, kind of through the body, and then pumped by a brachial heart into the gill, where it can be release its carbon dioxide, grab some new oxygen, return into the body. But there's other products that are being moved around in that hemolymph or in the blood of the cephalopod, and some of those are products of protein metabolism, so they are toxic. And within the cephalopods, that's where we see this little expansion. So this is the vena cava, so this vein that's coming in, and we've seen these kind of fingers fall down at the vena cava that's expanded to form renal appendages right there, almost these fingers, and they're in contact with, skip a little bit farther downstream, to the renal sac. And the renal sac, which is associated with the concentration, the creation for cephalopods of urine, those renal appendages are locations for the secondary reabsorption of, as that long protonephridial tubule, it allows for the selective absorption back into the body. But then the concentration of the urine through the nephridophore into the mantle cavity and then out of the body. And this is a focus, and I said it in 2700 about when we talked about mollusks, about how we need just a course on mollusks. And one of the associated, if we were going to have a course just on mollusks, we could have several different lectures about excretion strategies in mollusks, because they vary tremendously. They vary across marine, freshwater, terrestrial strategy. They vary across taxonomically, and so there is a large variation. So understand that what I'm talking about here is not, for example, a gastropod, a terrestrial gastropod system. It is not a bivalve system. This is the renal sac of the cephalopod, which is important enough to talk about. Well, why might I focus only on this strategy? Well, I picked it out of the molluscan amuse-bouche of excretion strategies, largely because the renal sac I find fascinating, because not only is it this place for the selective reabsorption of positives and the select of positive products that are of value to the cephalopod and for the concentration of the final ammonia-rich urine, but it is also the entire world for an entire phylum of organisms, of animals. So the the renal sac of cephalopods is the world for the dismidids. In fact, in the renal sac of one individual, you can find, or researchers have found, 2 to 3 dismid species, and most of those species tend to have a very high host specificity. So they do have a very complicated life history stage. There's a lot of arrows going back and forth, the vermiform, the infusiform, different embryonic forms that move into the sea and then parasitize different kinds, different lineages, different species of cephalopod. So there is a stage where they live the entirety of their life is the renal sac. There is also a stage where they are a very small component of the of plankton, of zooplankton, where they're moving around. They are excreted out. Some of these embryonic forms are excreted out into the mantle cavity, voided into the ocean, and then they must bump into, and these are unknown methods, so I'm scooching up to the video going, these are things that we don't know. They must happen because we see these host-specific associations across taxa, across different phyla, but we don't know how they find each other. We don't know that there's so much that we don't know at this level about an entire phylum of organisms. So if you like parasites, if you like marine systems, if you like excretion, if you like renal sacs, please go and become the dismid expert because the world needs you. So we're going to switch again, change the channel again. We're going to think about arthropods. In this case, one that you probably bumped into in 2700 when in your dissection or your consideration of the crayfish. And this is the antennal gland, what you might have heard it called the green gland, and that is, right here. Why is it called the green gland? It's not based on, it's not a surname, but it is just based on the color that you might have seen in dissection, that they form kind of a green color. And they appear a pair of glands that open at the base of the antenna underneath the eye here. And this is an excretory organ for aquatic crustaceans like your crayfish. And in this case, they are very important in the formation of primary urine that is ultra filtered. So in a freshwater organism, in a saltwater organism, getting rid of salts, not so important. There's not a huge difference in osmolarity across between the ocean, between you, unless you live brackishly, and we'll put that away. We'll come to it later. But in a freshwater system, there is a large osmotic difference between what you are internally, which is salty, and the external environment, which is not salty. So there's a whole bunch of your life which is described by keep the salt. And the green gland for the crustacean is a lot of how they do that. So that little squiggly diagram at the bottom here, this is a line diagram, a beautiful, I think kind of a beautiful one. And it makes me think of David Bowie because of the word labyrinth, but we'll put that aside for a second, where we see this is a green gland that has been teased apart. And so the green gland, that kind of amorphous green shape at the base of the antenna, this is what it looks like functionally if you spread it out, and this is the way to look at it, I think, in terms of figuring out what its function is. What is its function? We'll look at the plot on top. So we'll look at the concentration of salts in the liquid that it's along this gradient from the filtration sac, so this small sac or coelomic sac that runs into a wider region of labyrinth, which runs into a long tubule, a canal, which opens into a bladder. And you can see that along that pathway, we see a large amount of salt. The urine that's moving along that's the liquid that's being plotted there, becomes less and less salty. We see the reabsorption, the retention of lots of the salt. Because remember, this is a green gland that we dissected from an aquatic crayfish. The aquatic crayfish is playing evolutionarily a keep the salt game because the external environment doesn't have the salt. You need the salt because salt helps you fire and move and be you. And so the crayfish, the green gland, the antennal gland, is critical to the production of this hypotonic urine that allows them to eliminate water without losing ions. Switch the channel again. So we'll leave the crustacean and we'll go across to a chelicerate, and we'll look at a tick in this case. The tick, and so to orient yourself here, we have on the left-hand side of this diagram, we have like you or me. Some fleshy part that we didn't get covered with product or sock, and we've got a tick that has grabbed onto our tissue. And we see here the head of the tick that is eating, feeding on us. And we see two openings in passage of, the tick has latched onto a capillary gland, and we see the passage of some of our circulatory liquid fluid going into the gut of the tick. And then we see that moving into the tick, we see tons and tons and tons of sodium and potassium. And so most ticks that we would see around this part of the world eliminate most of their excretory water and those sodium and chloride ions by injecting it back into the host as they feed. So sodium from the blood meal is taken up from the midgut into the hemolymph and then pumped back into this salivary gland. And the salivary gland is then, as you trace the arrows, is moving it back into the host. So they're not excreting it across a diffuse membrane into the water. These are terrestrial organisms, but they're getting rid of those excess sodium and excess water by pushing it back towards the host during feeding. Now, another enormously successful taxon living terrestrially is, of course, the insects. And the insects, and we've talked about Malpighian tubules before, where you see a very reductionist view, a reductionist kind of excretory physiologist view of an organism here. So we see over here, not even labeled, the front end of the body with the face. So the face is over to the right and the anus is over to the left, and we see products moving through a midgut, and then where there's excess nitrogen and ions after digestion. But then we see these blind diverticula at the midgut and before the rectum, where we see cellular waste from metabolism that gets moved into the Malpighian tubules that then gets pushed out through the rectum and excreted from the body. So the number of this is a slightly more complicated view of it, but you see there's again the opportunity with these blind tubules. So in a dissection of an insect, the Malpighian tubules often look like kind of crazy white spaghetti. Lots and lots of them. They can vary. You can have one or two to hundreds, depending on the species of insect that you're considering. They end blindly in that body cavity, and so they are out there collecting all sorts of ions and low molecular weight solutes that are then we're reclaiming some of the compounds and lots of the water from. So this is a huge part. Water loss, if you're a terrestrial insect, is an enormous stress. And we'll talk more specifically about water loss. And we'll return and kind of expand on the Malpighian tubules next Monday. But we're building up uric acid here, and then we're excreting that as urine out through the anus. And this is the final button there is, bullet is what I'd mentioned earlier, which is we see it in the insects, but we see it in other terrestrial lineages that its most recent, their most recent common ancestor would not have possessed the Malpighian tubules. So this seems to be a trait, these blind spaghetti that diverticula of your digestive system is a really efficient strategy towards saving water and voiding nitrogenous byproducts of metabolism. Here's an even more classic diagram, and so we've got, and I love this one because we're kind of adding, we're still looking at a simplified drawing, but we at least acknowledge there's a head and a face and a brain. So we see an area where nutrients and water are ingested and they pass into the foregut, into the midgut, which is like essentially where a lot of the nutrients are being absorbed and the water. But then the Malpighian tubules here secreting the primary urine and being modified by selective reabsorption. That's the ionic kind of in and out diagram that you saw in the previous slide. That is then discharged and mixed into the gut contents before we move it out across into the rectum. And then the final bit of resorption that is happening here via anal papillae or perirectal tubules. So that's the little red dots there. Okay. I see some waiting room. All right. And no chat. So we continue. To consider, so what have we talked about? We've talked about 4 of the major players in the play. We've talked about guanine, urea, uric acid, and ammonia. We've talked about different organs and strategies that different taxa as a survey of excretory organs across the phylogeny of invertebrate animals, like coxal gland, the antennal gland, the green gland, the renal sac, nephridia, protonephridia. Some of the terms that we've used, that I've used already, I wanted to park this here before we talk about the in-the-world application of some of those taxa using those strategies. I want to make sure that we're talking about some of that we share a lexicon, that some of these words are not so deeply in your past as you're training as a biologist that we can consider them together. And so essentially we're unpacking osmosis, right? So osmolarity, so the amount of a solute or thing per volume of a solution, moles per liter. That we can talk about aquatic environments being hyperosmotic, isoosmotic, or hypoosmotic. And so that is facilitated by thinking about this cross-section of a container that has a semi-permeable membrane that has a concentrated side, the hyperosmotic side with a higher concentration of the solute and lower free oxygen or free water concentration. And then the opposite is the hypoosmotic side, where we see a lower solute concentration and a higher amount of water that's just on its own. And then from an organismal perspective, slightly, so certainly osmolarity and talking about these things is something that you have dealt with for a long time in your training, but perhaps you have not as frequently bumped into these 2 words describing what is not a binary, but is more of a continuum between organisms that are osmoconformers or osmoregulators. And so before we get to unpacking those, there's essentially, in a marine environment, we're talking about an isotonic environment. So salts and waters approximately equal, moving across a semi-permeable membrane, right? As opposed to freshwater, which is a hypotonic environment. So if you're an aquatic crayfish, water can move, but you're losing salts. Of course, you, the organism, you, the invertebrate, you, the organism, you are the oval. And then in a terrestrial, in a dry environment, it's hard in all kind of parts of your life because you're losing water and you're losing salts. Now, when we talk about conformers and regulators, we're going to consider them as the straw man kind of consideration. We'll talk about them as 2 different ends, as if they were separate, and then as we go along, we'll add detail to essentially realize that this is a continuum. So the conformer is the organism whose strategy is that their internal environment follows environmental conditions. And we've thought, or I've been mentioning salts a lot as we've talked about excretion, but this could be temperature as well, right? And a regulator is one that attempts to maintain a constant internal environment in the face of environmental conditions that vary. definitions, categorical definitions, but let's look at it on a continuous scale. Find me your annotation tool here and say, show me what a conformer would look like here. If you've got some variable on the x-axis, consider it salinity, from freshwater to very saline on the right, across an whatever their internal environment is. Show me what a conformer would look like. Yeah. Awesome. Perfect. All right. Now, I appreciate I'm going to clear them and say, you got it. That's exactly it. And we call that kind of linear relationship, we call it a line of conformity. And we're going to use it in our regulator consideration in a second, just as a way to orient ourselves in the graph, to say, if you were a conformer, you would fall on this line of conformity, this 1-to-1. But now I'm going to ask you to find your annotation tools again and say, if you're a regulator, what do you look like? Yeah, that's awesome. Awesome, everybody. That's great. So what you've drawn for a lot of you is a really strict regulator. So it's like it just maintains from whatever the highest and the lowest variation possible in that external environmental condition, be it salinity, for example. This organism needs to maintain an absolutely strict criteria. Now, some of you, where's one? Here's this line here. I trace. That is, let me clear these. Thank you for doing that. And say, and put up again that line of conformity that we talked about, that you identified from the conformers. But say, as a regulator, often at extremes, strategies fall apart. And so what we see is a period of hyper-regulation and a period of hypo-regulation, and then kind of forced conformity at either end. And this might be coincident with death or approaching death in these areas. That when you see these inflection points of hypo and hyper-regulation, where they can no longer maintain their internal environment at a different osmotic conditions than their external environment, their stress is high. Death, if not imminent, is likely. And if we take away the very simple kind of PowerPoint line drawing consideration of this environmental strategy and add back in this one, so we get the label of the osmolarity of the body fluid and the osmolarity of the environment, and then we're going to talk about the fact that this is a continuum. That is, it is not a binary. There's no perfect regulators. So the theoretical that you described all the way, we don't see that. We tend not to see it. And no perfect conformers, that conditions will always outstrip what capacity you have. So all animal taxa lie in between those two extremes, and they all experience some problem with the balance of water and of salts. So it's a continuum of strategies. So you drew out a perfect, strict osmoconformer, and you drew out, many of you, a perfect regulator. But you can see that we could just as easily talk about a strong regulator or a weak regulator or limited hyper and hypoosmoregulator. And so different strategies in areas of hyperregulation and hyperconformity associated with different parts, different potentially life history stages where they move through the different parts of the world, or different taxa, different close relatives. So we see here, and again, the X and the Y haven't changed here, but we've added some, instead of some descriptors of their physiological strategy, we've added some descriptors of taxa that really tend to do this, or environments where this strategy is necessary. So we see terrestrial insects and vertebrates. They tend to exhibit this kind of hyporegulation because losing salts and waters, both of them, is very bad. brine pool mosquito larvae. So saltwater, they have this little bit of an up and down at the other end, but they're a regulator across a wide strategy. Then we see marine environment, elasmobranchs or estuarine crabs that live in estuaries, annelids that live in estuaries, or a prawn that's moving around, I would presume, across some gradient of fresh to salt water. And then we see out here in this little, I'm going to call it again a chocolate chip cookie, but I suppose because I'm hungry, but most marine vertebrates living out in the exterior end of being really regulating it. So we see descriptions then of where they sit in that space, but then descriptions in text here with some of their derivation of the words on the right-hand side. So a stenohaline in organism, so like this brine pool mosquito larva. This is one that can, sorry, it's not that one. The stenohaline, so it's narrow. So steno means narrow. So that would be these guys, right? Like terrestrial insects and vertebrates, or the most marine invertebrates. So in brackish water, we've got fluctuations in salinity. So that's happening as a river flows into the ocean, for example. So Uri is Greek for broad. Hals is still salt. So there's some osmotic regulation. So like an estuarine crab up here, where there's some regulation and then the assumption of a more conforming lifestyle. So in stenohaline invertebrates, the body surface is responsible for most of that salt loss and uptake. And most crustaceans that live in salt water are stenohaline conformers and have some large range of tolerance. And that's important because when we look at a river, so you're now a satellite kind of looking down at a really simplified river kind of belching out into an estuary. So freshwater moving this way, and you can see 5, 10, 15, this is the salt wedge. So very saline out in the ocean or the sea, and less saline as you move upstream. And if you're an organism that lives in this kind of brackish estuary, or that is small enough that it can't control a lot of its movements, that you get moved with the water, you're going to experience the salt wedge. You're going to have to live across that variation. So complicated diagram here, but there's a lot of good truths in it. So let me don't look at the diagram yet. So what we see, let's look at the top half, is the the river is still approaching from the left-hand side. And we see descriptions of limnetic organisms living in the river, oligohaline, meso, poly, and euhaline. So from the head of the river up into the mouth and into the ocean, we're looking at an increase in the change to full parts per million of salt water. And then we see different descriptions, ecological classifications of organisms across that variation. So there's stenohaline marine, the ones that can live only in the salty conditions. And if you're a mixohaline, you have an incredible range of strategies because you can go upriver into the freshwater, you can go downriver into the highly saline. So the ecological classification there depends on, we're codifying it based on where they spend all or part of their life cycle along that salinity gradient. And then we've got on the bottom a plot of across that osmolarity from fresh to salt to fully saline of species richness. So we see an increasing diversity of marine organisms the farther out. This makes intuitive sense that you don't see a high diversity of marine organisms in a river-based system. But we see freshwater organisms, highly diverse and falling as you move closer towards the sea. Brackish organisms, they are most diverse in that intermediate stage. So in a continuum of freshwater to marine systems, water from the environment living as an aquatic organism is a challenge. And we'd say right back to that simplified oval that aquatic is more challenging than marine. Terrestrial is perhaps more aquatic from these perspectives than terrestrial is more stressful, challenging than aquatic or marine. But if you're an aquatic organism for whatever stage of its life history, whether if you're a dragonfly living there as a larva, or if you're just living there your entire time as a crayfish, you're gaining water, you're gaining all sorts of things on the right-hand side, but you're losing, there's loss of salt from your salt glands. There's lots of salts from ions and water from your gills, from your cuticle, from your eggs and gametes, from your excretion. And so there needs to be, since water from the environment is constantly entering the tissues, they either have to pump the water consistently out and or they need to regulate solutes and fight the loss of the salt. And so most marine invertebrates are these osmoconformers, where if we look at where they live as cnidaria or as cuttlefish, that their concentrations of their plasma, of their body fluid, as a percentage compared to seawater, is very nearly the same. There's variations, of course, across taxa, but we see largely just osmoconformation. And so for many of these marine invertebrates, if we look across this osmolarity continuum, you'll see a high diversity here with some regulation and then some conforming, but into all of these, so the Artemia is a fish, but it exhibits it's on the graph because it has a wide, the most brackish fish. But we see not a whole bunch of invertebrate taxa that can show you that nearly strict regulation. And in fact, this example here, the most apparent regulator of all the invertebrate taxa, is now not even considered to be a single species. It's not even a monophyletic group of Nemertina. This is a ribbon worm. So it's most likely many species. And so if I share that with you, consider what you might have to do to this, our consideration of this as one unit that has evident strong regulatory powers, or is it if it is multiple taxa, do we see slicing of this, and each one of those taxa operating across only a smaller component of that osmotic challenge? So within freshwater invertebrates, they're all regulators to some degree, but they all have a continuous problem with water balance and a reduced permeability, and they have ion uptake mechanisms with regulated hypoosmotic. So we see freshwater fish here with the specialized migratory fish that have the regulatory capacity to live across that gradient. But most of the freshwater invertebrates we see sit here above the osmotic line of full conformity and exhibiting hyper or strong regulation. So starting back with our earworm from Brittany 40 minutes, 45 minutes ago, 50 minutes ago, this is where we've been. We've talked about the four major players, some different excretory organs, some shared terminology, and then the distribution of a lot of these organisms, particularly across an estuary or a brackish continuum. And that's about it for our time today. What I'd like to do next time, next week we're going to talk about water loss or water stress and temperature stress. Next week you're in the lab, you're going to keep on keeping on with your lab experiment. What to work on, the case study is going to come out on Thursday and you have to finish it by Monday. And then of course this week you have to finish your invertebrate R assignment and submit it before midnight on Friday. And a heads up that I am traveling again this weekend, so I'm not going to be able to respond rapidly to questions about case studies or invertebrate R by email or discussion board from Friday until early Monday morning. So finish up the invertebrate R, enjoy, and take care of you, take care of yourselves, take care of others around you. I'm going to stop recording and hang around if there are any questions.